Hey team, Dr. Jack Order here, and in the next few videos, I'm going to be taking you through viruses and viruses as pathogens, because we can't talk about diseases without covering viruses. Now, interestingly, this a playlist is called the cellular basis of disease, and as we might learn, viruses, are they really cells? But we have to cover them because we've got to talk about viruses if we're going to talk about diseases. They are one of the most important and relevant today. So we're just going to start with a little introduction, and as my little background image there, that is the one and only SARS-CoV-2, aka COVID-19 virus. And we can see why it's called a coronavirus, because it looks like it's wearing a crown. That's where that name comes from. And these are those infamous spike proteins which you might have heard about because of their role in infection and also the role in creating vaccines against them. But I'm getting way ahead of myself. Let's rewind and go back to the beginning. The first thing you've got to understand about viruses is that they are tiny, incredibly tiny. Even on a microscopic scale, they are tiny. So here we have a mammalian cell, here we have an E. coli cell, and here we have a SARS-CoV-2 cell. Now, obviously, I've zoomed in for all of these. Now, I'm going to try and scale them appropriately. This is roughly them scaled again. Now, actually, I couldn't quite scale it quite right because I can't actually make it small enough in PowerPoint. Viruses are absolutely pathetically tiny, and they're pathetically not complex. Let me demonstrate how non-complex. So I've copied and pasted the human genome into a Word document. So this is the A's, T's, C's, and G's that make up our DNA. There's about 3 billion of them. 3 billion of them. And if I read it out going at 1 a second for 24 hours a day, each one of those letters, it would take me over 120 years, over 140 years, I think, to read out the entire human genome, even going 24 hours a day. I couldn't even fit the entire human genome in a Word document. I kept crashing my computer. So this, I think, is about a third of it and is 25,000 pages long. Right, now let's compare that to the SARS-CoV-2 genome. Copied and pasted that into a Word document. To read this one out, it would take around about 6 hours. And as you can see, we can even just fit it on a screen. It's around about 25,000 nucleotides. So it is so incredibly simple. You know, their number of genes, the number of chunks of DNA that actually do something, number in the single digit, somewhere between 5 and 11. Okay, so maybe just into double digits. Whereas we have about 20,000 protein coding genes and so many more if you start to look for functional units of DNA, depending on your definition of a gene. So they are incredibly not complex. How do they get away with being so uncomplex, so incomplex? How do they get away with that? So there's a lot of variation within viruses, first of all. So it's hard to talk about them generally, but all viruses have a container, and in that container, they have a genome. And then, honestly, not too much else. Now, this virus is resting up against a eukaryote membrane, so a membrane of one of our cells, and this is inside the cell. This is the abbreviated life cycle of most viruses. They either fuse with the membrane or they get inside our cells and crack open, and they release their genome into the cytosol. The genome then hijacks the cell's biology to read the genomic instructions and build more viral particles. So the virus doesn't need the genes for a ribosome, because the cell's ribosome is going to make the viral proteins for them. The viral genome does not need the genes for metabolism because it is not consuming or eating anything or digesting anything. It is stealing the energy molecules from within the cell itself. So the cell's doing it for it. It doesn't need any of the enzymes involved in lipid generation because it's going to steal the lipids from the cell. So this is how it avoids all its complexities. It avoids the necessary complexity for life. And it actually brings out a very important question. Are viruses alive? And it's a bit of a tough question here, because when a virus is a particle. It's a viral particle. When it is an entity, a something, it does nothing. It literally does nothing. Okay, there's no metabolism going on, there's no consumption of anything, there's no replication. When it is an entity, it does nothing. It's only once it breaks open and releases its contents and stops becoming an entity does anything actually happen related to the virus. And then it relies completely on something else to generate, like a mammalian cell, to generate more of these particles, and then those particles go off. So it's hard to say that this meets the minimum threshold of life, because when it is something, it doesn't do anything. It's as inanimate as this pen. It doesn't do anything when it's by itself. And it's only once it breaks open and becomes a milieu of chemicals does something happen. And it's hard to call something like that alive. So it's in a real gray area of biology. And it becomes hard to even talk about it in microbiology. Is it a microorganism? But it's not an organism, because part of the definition of an organism is alive. So it's a tough and interesting question. So how do we break up viruses? How do we talk about it? I mentioned that there's a lot of diversity in viruses, so we need to break them up into chunks so we can actually talk about them. We break them up a few ways. First of all, the genetic material. So all eukaryote cells have a DNA genome. However, viruses have either a DNA genome or they can have an RNA genome, which is super interesting. SARS-CoV-2 is in that category. Or they can have a single strand of DNA, which is even also interesting, right? So it's now just a single strand of DNA instead of the typical double. So we can break them up by that, by what kind of genomes they've got. We can also break them up by that container. There's two main groups. There are others, but there's two main ones. There is a protein capsid, which is a rigid structure made entirely out of proteins. And then there's a phospholipid envelope, which is much more like our membranes. It contains those phospholipid bilayer, and it contains proteins. Now, it's much more like our membrane because it stole our membrane. That's why it's like our membrane, because it is our membrane. It has been stolen by the viral particle. So those are two of the main structures, that main container, and it's another way to divide up the viruses. There's also the classic genetic similarity, and from this we can attempt to construct species. The term species kind of breaks down at this point, but we base it off genetic similarities and evolutionary proximity. And beyond species, there is also types. Now, types is a bit of a vague term, but when we're talking about viruses, what we mean by types is something that our immune system would say is different. So our immune system's always trying to detect pathogens, and it detects viruses by what's on the surface of that virus. And so if our immune system, say we get infected with one cold virus, let's go influenza. We get infected with one influenza, then we get infected with a different type of influenza. Our body won't remember that different type of influenza because the proteins on the surface of that influenza have changed enough that our immune system can't recognize it. So we define types by our ability to recognize them. As our immune system, right? So this is why we need a flu vaccine every year, because the type has changed, right? Okay. And also life cycle. So I gave you a very simple life cycle, but there is actually some crazy complexity when it comes to viral life cycles. In fact, I'm only going to touch on one because there's so many different versions, and they're all brilliant but complex that we don't just want to go learning a whole bunch of life cycles. And if you actually stick with me and subscribe to the channel, I'll definitely cover some more complicated ones in the future. Okay, so as an example of how we break up viruses, I'm going to go through the common cold. Now, the common cold is mostly caused by a virus called the rhinoviruses, a group of viruses called the rhinoviruses. Now, this is very easy to remember. Rhinoceroses have that large nose, don't they? Well, the horn. And if you get plastic surgery on your nose, it's called rhinoplasty. And so a rhinovirus just infects your nose, right? It infects your horn, if you will. Now, A rhinovirus has a protein capsid, so we can put it in that group by its container. It has an RNA genome, that's another group we can put in it. Now, it has 3 species, which basically means the genetic diversity is enough to define them as different species, but over 160 types. This is why you don't get immunized for the common cold and why we're just constantly getting the cold over and over again. It's because there's so many different types of rhinoviruses. Now, the rhinoviruses is 30 to 80%, cover about 30 to 80% of the common cold. So what are those other non-rhinovirus common cold viruses? Well, another one is the adenovirus. Now, it has a protein capsid. Again, very cool geometric structures here. Icosahedral, we call these. Very, very cool shapes. It has a DNA genome. There's 8 species, 2 of which cause cold-like symptoms. There's a whole bunch of others that cause, the other ones often cause things like intestinal diseases. There's loads of types, 88 plus types, and it's responsible for 5 to 10% of common colds. One of the reasons why I wanted to introduce this guy is we use adenoviruses in research all the time. It's an amazing tool to get genetic material into cells that we want to get genetic material cells into. So it's a genetic material delivery system that we utilize. And in fact, the AstraZeneca vaccine for SARS-CoV-2 is an adenovirus, and it administers a SARS-CoV-2 gene into the cells, which allows them to express an antigen, a SARS-CoV-2. I'm getting way too ahead of myself into the immune system. But anyway, it shows the body a small chunk of the SARS-CoV-2 genome, which leads to a small chunk of the SARS-CoV-2 protein. But boy, I almost got carried away into the full thing. I'd love to tell you more about it, the AstraZeneca vaccine and the adenoviruses. But the last one that causes the common cold are coronaviruses. What this means is you, listener, have probably had a coronavirus and hopefully haven't had SARS-CoV-2. I'm really sorry if you have had SARS-CoV-2, but if you've had many colds throughout your life, a small percentage of those are coronaviruses. Now, these guys don't have a protein capsid. They've got that phospholipid membrane, and in that membrane are those infamous spike proteins there that we can see. It has an RNA genome. There are many species, 5 of which cause the common cold, and we are now aware of another species of the coronavirus, SARS-CoV-2. There are many types. We're still counting. It's endless when it comes to these types, and it's responsible for 5 to 15% of colds. So here we just ran through some viruses to see how we would break them up based on those, the container, the genome, and things like species and type. So where do they sit on the tree of life? Well, I mentioned this. We don't really class them as alive. So they're certainly not prokaryote or eukaryote, and it's hard to place them in the tree of life. So where did they come from? Now, this is still highly contentious. There's 3 main classical theories about where they come from, but there are actually even more bubbling up. But I want to take you through these 3 classic ones because it's a great way to explore evolutionary ideas and where pathogens sit amongst the tree of life. So the first one is the virus first hypothesis, then there's the escape hypothesis, and then there's the reduction hypothesis. And these are all absolutely fantastic. So let me jump through it. So the virus first hypothesis is the idea that sort of the first organic entity was a virus life form. Now, they then branched off and some of them remained virus, and the entire viral family tree came through one of the branches of this original virus, but another branch gained complexity. Now, I find this one tough, but there's very reputable viral scientists that believe this theory, so I'm sure there's just some arguments that I'm not aware of. But I just think the fact that viruses are obligate pathogens makes this a tough pill to swallow. Now, there's the escape hypothesis. Now, I actually, personally, this is my favorite. And if I was to put my money down, this is where I'm going. It's the idea that some biological process in a more complex life form, probably a eukaryote more than a prokaryote, accidentally created the first virus. And then the first virus then replicated and mutated and evolved and gave rise to all the other viruses. So how could a eukaryote cell give rise to a virus? Well, remember, eukaryote cells are full of RNA, right? So maybe it was an RNA virus that was the first one. We full our cytosol. Our entire cytosol is full of RNA. That RNA codes for things like polymerases, which can replicate genomic information. Also, the genes for envelopes for developing membranes around, for manipulating membranes into balls and stuff like that. So yeah, what if we accidentally blebbed off a virus and then that virus became infectious and out of control and our bodies didn't have the ability to regulate it, and then we started shedding this virus and it evolved into the full viral family tree? Now, let me show you an image that might convince you of this. This is an electron microscope image of a eukaryote cell. And what it's doing is it's blebbing these things called microvesicles. So microvesicles are just blebbing off the membrane. We can see them there in these little balls there. Now, one of the functions of this in the human body is we pack them full of microRNAs, these very small RNAs, and then the vesicle then goes fuses with another cell and injects those microRNAs into the other cell. Those microRNAs can then turn off genes. So it's a way for one cell to regulate the genes of another cell. But you can see a lot of the viral life cycle in that process. If we bleb off a bit of membrane that contains small bits of RNA that's designed to interfere with the biology of another cell, that seems to me only to be a few tweaks away from a virus, accidentally creating a virus. I also come from plant biology, and there's these crazy things where there are virus-like entities that can't leave the cell, so they just replicate inside one cell and they have no ability of getting out. And so it's only when the cell divides can it get anywhere. And because plants propagate vegetatively, you know, they can just shoot out a root and another plant grow. That's kind of how the virus spreads, but that to me just sounds like like a cellular process gone awry that we can't turn off, almost like an internal cell cancer rather than a cellular cancer, a cancer of the cell, of the processes going inside the cell. And so that convinces me again of the escape hypothesis. I quite like it. Then the other one is the reduction hypothesis. I think this is definitely the second most likely. So it's the idea that life had a common ancestor and it broke off and we ended up with archaea and bacteria, and then that eventually turned into the tree of life, the eukaryote tree. But some lost complexity, and so this is what the reduction is. We got a reduction in complexity and a reduction of independence. And so the common ancestor of all viruses is not a virus, but we got this reduction in complexity and this over-dependence, and we ended up with our first viruses. So those are the three main hypotheses. You can tell I like two of them. But if you're out there and you're a virologist and you like the if you like the virus first hypothesis, hit me up. I'd love to know the details on why you think that that's a good theory. Now, communicable or non-communicable, obviously it's communicable viruses, right? They're one of the easiest to spread diseases in the world. Yeah, so it's a communicable disease. It's not one of those cancers or dementias that are non-communicable diseases. So are they a clade? Now, that's a really important question, right? So if we look at this tree of life here, it certainly looks like a clade. A clade, remember, is a branch of the tree of life that you can snip off and all the members would be contained in that branch. So if we snip the branch of the tree of life there, you can see that all the members of the virus would be in that clade, right? So are they a clade? This depends on which theory you believe, though. So with the virus first hypothesis, it seems clear that they are a clade because they are one of the branches of the trees, and viruses was the first branch, essentially. With the reduction hypothesis, again, we got that branching off, and then we got the branching of the branch, and so now we've got this big trunk that has a common ancestor of all viruses. So the reduction hypothesis, they are a clade too. The escape hypothesis is the answer is Probably, but maybe not. So maybe they escaped multiple times. So perhaps a protist released a one kind of virus, and a bacteria accidentally released a different kind of virus, and a perhaps even more complex eukaryote cells beyond the protists, perhaps even multicellular organisms, shedded viruses. So in this way, you could see that there might have been multiple evolutions of a virus. And in that case, they grew from separate branches, right? The viruses appeared and grew from separate branches. And in that case, they would not be a clade. But maybe they only escaped once. So it doesn't go either way. We're not sure if we believe in the escape hypothesis. But the reason why we're interested in clades when we talk about medical conditions and medical risk and medical management is because of selective toxicity. Because bacteria are a clade, they're genetically similar, so they contain proteins that are similar, so we can target those proteins to kill all bacteria, right? Because bacteria are a clade, we can create antibacterials, right? Whereas protists aren't a clade, we can't create a general anti-protist drug. It has to be specifically designed for each tiny subgroup of protists because they're not a clade. So if we look at viruses, the answer is that they're probably a clade. You know, most theories agree that, you know, there's a high probability that they're a clade. But the problem with viruses is that they're so prone to mutation, they are so genetically diverse that targeting them through selective toxicity and treating them like a clade is incredibly difficult, right? And so this is why we don't have a general antiviral compound, right? Because they're so diverse. I've tried to illustrate that with this giant branching of the tree there. The genetic similarity between these viruses is so low that they don't have proteins that are similar. And that's because of these incredibly high mutation rates. Why do they have high mutation rates? Well, our cells contain DNA checking and DNA repair mechanisms, but they're in the nucleus, so they're not checking and repairing the viral genome, and the viral genome does not contain that. Also, a lot of viral genomes are single-stranded, so they don't have another strand to check if a mutation occurs. Also, viruses have a very short lifespan, so they are constantly being created and undergoing generation after generation after generation, which evolutionarily speaking, accelerates the mutation rates and accelerates evolution, which means that they diverge incredibly quickly. And we're going through this. We're up to the Delta variant with the coronavirus, so we can see how quickly they mutate and change. And so they're such a diverse group that targeting them with a general selective toxicity, because we can treat them like a clade, doesn't quite work. Another problem is horizontal gene transfer. So some of our genetic material has come from viruses. I'm not going to dive into this hugely, but some viruses insert their genome into our genome, and on occasion it can get into our germline, so our sperms or our eggs, our gametes. And so we then have viral genes in our genome, and so it's hard to, and vice versa. Some of our genes are in viral genomes, and this happens across the tree of life. So then if you want to say, is this virus more related to this virus, when you start to do those genetic similarity tests, you get incredibly weird results because some of their genes look more like our genes than another virus's genes. So again, this creates massive genetic diversity, and it also makes their evolution very difficult to follow. We know we're more closely related to a dog because genetically we're closer to a dog than we are to a fish, right? So that genetic similarity allows us to know that we branched off in evolution more recently than we did with fish. With dogs. So we are more closely related to dogs because we can prove that through genetic similarity. We can't do that with viruses because they stole some of our genes and we gave them some of our genes, or you know, across the entire tree of life, not just us. So in summary, wow, that was a big one. They are pathogens because they will go into this, but they're obligate parasites. They have to get into us. We don't typically call them parasites because they're not alive, they're not organisms, but they're obligate pathogens. They are probably a clade, but targeting them through selective toxicity by targeting genetic similarity is not likely. So that's the summary of the introduction of viruses. Up next, we're going to go over viruses as pathogens.